Welcome to the Medical Device Made Easy podcast. Here is Munir Lazouzi from easymedicaldevice.com. And today we'll try to help you to master your biological evaluation reports or biological evaluation testing that you will be doing. And we'll try to help you to target or to understand what are the top mistakes that are done within this kind of evaluation. And for that, I have two peoples from the Groupe ICARE in France. So we have Laura Fusari and Paul Fernandez. So Laura and Paul, welcome to the Medical Device Made Easy podcast.
Hello.
Hello, thank you.
Thank you for having us.
You're welcome. So just as a reminder, we had previously also the Groupe ICARE with Philippe Bourbon. Last time we discussed about a clean room, so how to set up a clean room, how to monitor a clean room, everything. So if you are still interested about clean room, you can go to this episode again to look at that. So today with Laura and Paul, we'll discuss specifically about biological evaluation. So Groupe ICARE is a laboratory that is helping, if I can say, it's customer related to tests that to be done. So I think they have a lot of experience to share with us. But first, so just maybe a small introduction from yourself. So what exactly you are doing at Groupe ICARE, both of you?
So I am, and Paul is also a medical device safety and toxicological safety assessor. We guide clients through the biological safety assessment process. And for that, I did, before entering Groupe ICARE, a master degree in toxicology.
Great. And you, Paul, the same?
The same as well, master degree in toxicology, and now I'm a risk assessor for Groupe ICARE since 4 years now.
Great. So I think, as I've said, you have some customers, so you are meeting with some people, you are reviewing some documents, you are doing all those things. So I suppose that you have a lot of, if I can say, experience in terms of guiding, helping, showing the right way, and maybe also trying to avoid some mistakes with those customers.
Yeah, sure. It's our main goal in our job every day. It's to guide correctly people and clients and to try to avoid the mistakes we are going to talk about today. These are maybe obvious for some listener of this podcast, but if we saw so many times these mistakes, it's important for us to share our experience and what we have done in the past with our clients.
Great. Just maybe as an introduction, because we have sometimes some people in the audience that are also not maybe familiar with what is a biological evaluation and what it means exactly. So if we ask you in 1 minute, if I can say, to tell us what is biological evaluation, how we can, why do we need that maybe, and how people should be complying to it. So how would you do that?
I would say that it's a multi-process thing that manufacturers are using to demonstrate that the medical device is biocompatible, which means that a medical device will work correctly as per the intended use in the patient without creating any adverse effects. And the best way to assess for biological evaluation is to use regulation, so the MDR, and standards for biocompatibility, which are the standard 10/1993.
Yeah, the famous standard, if I can say so, it's not only one standard, there are multiple standards out of that. So we have the 10993 -1 that is mainly the main one to look at. And I'm sure a lot of people have looked mainly at this one because there is this famous table that is telling you exactly which test to do if your product is in contact with your with the patients or not, and the time of contact, and invasive or not, etc. So then it tells you exactly what to do. So I'm sure a lot of people have seen this one. But behind that, there are a lot of other standards that are explaining how to make those tests also happening there. So so one thing also important, do every medical device need this biological evaluation, or we have maybe some medical devices that don't need that at all?
I would say that all medical devices need to perform this biological assessment, and then within it, you have to show that you can fulfill all requirements if you have a critical medical device. But if you have a less critical medical device, but we will talk about it maybe later, then you will still have to perform the biological evaluation, but it will be lighter.
But so, for example, let's say a software doesn't need an biological evaluation. It's not touching the patient, but or there is still a biological assessment to be done for a software, for example.
For a software, it's an exception, of course. The software will not touch the patient, but if you follow, I would say, you follow the MDR. The MDR tells you that you have to ensure the biological safety of your product if it's a medical device. Software being a medical device, when you're going to check for this point, the biological safety, the answer will be very easy. There is no contact with the patient. It's a software. It's digital things. The evaluation stops here. You just have to say that no contact will occur with the patient, and so no risk, no toxicity can be applied to the patient because of this software. So a software don't need a biological assessment, a complete biological assessment, but you have to justify why.
Exactly, yeah.
And the process is based on risk analysis, so you still have to highlight risk and respond to them and show that you don't have probability to expect them, or if they are present, that they are here.
Exactly. So mainly, yeah, the idea is to say that you have to follow all what is written on the EU MDR, but we have also to have some kind of common sense and to say, yes, you need just to justify that you don't need it. It's mainly that's explain to me when you did. Like for people that are maybe used to do the GSPR, you have a lot of elements in the GSPR that says not applicable, but notified bodies are saying, okay, it's not applicable. Explain to me why. Because maybe it's obvious for you, but for us who is making the evaluation, we don't know. So explain us why it's not really applicable for that. So today we plan to, if I can say, educate the audience about the top mistakes that are done when we are doing a biological evaluation. So if I remember, there was around 7 mistakes that you have told me. So let's review them one by one, and maybe we can evaluate if there is any question in the meantime. So those mistakes are maybe mistakes that you have noticed while you are doing this exercise with your customers, or where you are doing your biological evaluation with the customers.
Yeah. Go ahead.
I think the most important part is when manufacturers do not begin or do not assess correctly duration and nature of contact. Like we said before, basis for biological evaluation is the standard 10/1993-1. It sets a list of endpoints to fulfill, and it depends on the criticability of your medical device. And for example, for short-term medical device, for medical devices that are in short-term contact with the patient, you have an amount of requirement to provide that is less important than an implantable medical device. And some points are sometimes missed by manufacturers. For example, the standard states something that I think is interesting. It's a transitory contact medical device, which are medical devices that are in contact with the patient with a duration inferior to 1 minute.
Exactly, yeah.
And for this medical device, sometimes you don't you are not required to perform biocompatibility testing. On the contrary, there are manufacturers that think that their medical device has a limited contact time with the patient, but this contact is repeated for a certain period of time, and in final, it sets a larger duration of contact, and so endpoints are longer.
No, it's clear, and this reminds me when you are doing the classification of a medical device, it says, for example, a transient use is below 60 minutes, if I remember. Then short-term use is between 60 minutes and 30 days, and more than 30 days is long-term use. And I know that some manufacturers say, okay, my device should be used during 50 minutes, so 50, and then replaced by the same device, and they think that it's just one-time use. But here we talk about cumulative use, and here is the same. So it's really cumulative use that is really important. For this contact time that we have, so we're talking a lot about the patients. Are we also talking about the user, like the surgeon or this kind of thing, or not at all for this kind of products?
Yeah, if we talk about the person that is in contact with the medical device. So if you talk about a gown, for example, it's for a surgeon. They are the users, but they are, in this case, the patients.
And so you need to do the test. For example, a nurse that is manipulating a device each time. It's a medical device. It's then placed in a room. So we manipulate the device multiple times. So for example, the interface where there is a buttons, etc., there is some silicones or whatever. Do you have also to assess that as it's touching the nurse, you have also to have some biocompatibility with that? Or you can say, no, it's a common material that is used by anyone, even outside of medical devices. So then there is no need of making this biological evaluation.
You need to make the biological evaluation. Like we said earlier, it's a medical device. You have to do your evaluation of biological risk. The nurse in question, if the nurse touched the interface, risk may be, if we extrapolate, maybe an irritation reaction on this end, sensitization, allergy to material used to make the interface. You have to justify that these risks are managed or totally absent of existence for the device. You can perform tests if you want, but you can just, based on documentation, based on the knowledge of your material, the origin of this material, if they are maybe of medical grade, it's not, I would say that, it's not the only thing that will be used to prove your biocompatibility, but all these are arguments you can use to prove that there is no risk and avoid testing. For such device, the requirements of ISO 10993-2, which tends to limit the animal testing, to use in vitro testing as much as possible, go in this way. Your device is not a critical medical device. It's not implanted. It's used for a few amounts of time, even if it's repeated along the time. Risks are easy to identify and sometimes really easy to manage only using your knowledge of the material. You don't need necessarily to conduct testing on an interface of an in vitro diagnostic medical device.
Okay, I think it's great, and yeah. So mainly this first kind of mistake of the time of contact is mainly. Is it like something that you see a lot, or it's just really rare that this is happening?
No, it's rare. It's
Okay.
not that common. Manufacturers tend to have set the duration and nature of contact before entering the biological evaluation process.
And I suppose there are some specific devices that are really exceptional, and sometimes it's really difficult to identify those timings. Yeah.
It can be the case when you work with companies that are manufacturing a fresh medical device, an innovative medical device, which can take part of device already existing. So this part, you know how to classify it according to ISO 10993. But some parts are in contact in a different way or in a shorter way, or maybe this part is an accessory that you have to treat like a medical device if we we mentioned the NDR. So sometime in some case, you will have to classify different parts differently, okay. and then assess each part as it is classified by the standard. And not, it's something I saw a few times. You have the most critical part needs it's implantable. You need to do many tests, and so the manufacturer tells us we can take all the parts and make all the tests on all the parts.
Okay.
All right, if you want, but first you will maybe go to animal testing that was not necessary for some parts, and it's a requirement of the MDR and of the standard to limit as much as possible animal testing. And on the other way, it will cost you much more if you do a really long test on each part of the material. We are scientists at the beginning, and for us, the scientific reflection is really important, but we are conscious that our client, our manufacturer, have money issue, or budget is really important thing. The economy is not very well in all countries. We have also to have this in mind to guide them for the most satisfactory strategy.
Most efficient.
Most efficient strategy in terms of scientific value, in terms of economic value.
No, I think it makes completely sense, and and this is also the idea here is also that the laboratories can help you and guide you in the right direction instead of you just thinking of everything. Just having maybe a discussion with your laboratory can really help you. The second issue, what is it?
The second issue, I'm looking for my note. Yes, the data collection.
Okay.
Maybe Laura, you can start with this.
Sometimes we saw manufacturers that think, like we said, that biological testing and biological assessment, that biological assessment is biological testing only.
Okay.
In fact, the most obvious part to begin with is data collection, because if you have a medical device and raw materials, then you have suppliers that have data on this raw material. And with this data, we can need literature review. So combining supplier data and literature review can help in the assessment. For some medical device, it can be the complete evaluation, and for other, it will guide to see what are the next steps.
So when we say data collection, it's prior to testing or after testing, or what's exactly your idea of data collection?
It's completely prior to testing. It can even allow to avoid testing.
Okay.
It's just analyze all the data you have in hand about your medical device that can set a proper identification of your medical device, which means go through a listing of all raw materials, the quantity, search in literature review if these raw materials are known to be toxic for users, for animals. It's the first step.
Are used in similar devices. Yeah. We can find, it's really rare to find specific data in the literature on a device we are evaluating here in ICARE. But it can be possible, or if you are looking for information on stainless steel, we'll have a lot of publication on stainless steel, and it's used, okay, in industry, but it's used in medical application. It's known biocompatibility. And to go back on a point we mentioned earlier, you can choose the material to make your medical device by using criteria that material is known to be biocompatible. The manufacturer or the supplier of this material said that he used a biocompatible, he manufactured a biocompatible material, and the compilation of this data can be a really good starting point on the biological evaluation before any test.
But I see also that some manufacturers are using just this information of initial to say, my material is biocompatible, so I don't need to do a biocompatibility test. So what do you say to that?
It's a common mistake.
Yeah.
I will not say it's really a mistake, but it's an idea many clients we have in mind, and it's not correct. If a raw material supplier claims an USP class 6 or a biocompatibility according to ISO 10993, that's great. That's obviously great, but this do not take in account manufacturing process, cleaning, sterilization, contact with other material in your device. If you buy silicone for your medical device, your device may be not completely in silicone, and interaction between two types of polymer or material can impair the proof of biocompatibility the supplier perform or prove in the past. So it's a good point of departure. It will help us as assessor to identify the most critical toxicological risk and some that we may be discarded because silicon is medical grade, for example, but it's not an absolute proof of biocompatibility of your final device. And that's a mistake we saw often, people coming to us with an implantable device saying that it's a titanium prosthesis, it's well known, it's used for many years. Okay, not a problem. We have data on titanium, we have feedback on such device, but your process may alter the material. Have you any proof of its durability in the patient body for 30, 20 years? I don't know. All of the things that put your device in a real context, in a clinical context, are not taken into account by the manufacturer of the raw material.
Exactly. As you mentioned, so the oils that you are using for manufacturing or the heating, the process that is maybe modifying the structure or all those things you cannot copy that from another manufacturer, maybe because it's something that is really proper to your company. So which is difficult. But as you say, it's a good start, but mainly this is not something that is possible. So I hope this will help all the manufacturers that are listening to say, don't just say, oh, it's a biocompatible material, so my product is biocompatible. No, it's there is also some analysis to do. So just maybe to continue on this one. So if the material is biocompatible, there is less tests to do, or there is the same amount of tests to do?
It depends if you know properly your manufacturing process.
Okay.
As you said, if you can trace all the oils, all the cleaning agents that are used during manufacturing process, and your raw materials are biocompatible, then depending on the contact of your medical device with the patient, it can be the end of your biological evaluation process.
Okay.
In most of the cases, it's not the case.
Yeah.
And thus, you have to complete what we are talking about, and which is a step called the chemical characterization. And you have to do testing. So you use the extraction process, which is you take your device, you put it in a fluid, and you identify and quantify all the substances that can leach from your medical device. And that is the second step to complete the biological evaluation process. Based on this step and based on calculation you can do with the result you obtain, then you can have your biological evaluation process finished, or you have to complete with testing.
So as you said, the collection of data prior to do the test is really critical because it can help you to reduce dramatically the number of tests, or identify maybe something that can be helpful for you for doing that. So I think it's one point that is really important here. What is the next one? Next mistake that people are doing?
So we talk about data collection, data generation with the chemical characterization. It's a generation of new data missing, and then
not choosing the good test element.
Okay.
So what the standards say is that the test item should be in the same final form as your medical device and at what the user is exposed to.
Okay.
Based on that, you have several strategies we can see from manufacturers. Some choose to test the final device. It's the easy way.
Okay.
But for some medical devices that are way too big, for example, to be contained in the vial I was talking about for chemical characterization and the fluid, then you have to do a representative sample. But whatever the case you choose, if you want to test your final device or a representative sample, it will have to pass, and I think it's the most important thing because sometimes it is missed, it will have to pass all the manufacturing steps, including sterilization, in the same way your medical device is supposed to.
Hey, just a second. Do you need a EU, Swiss or UK representative? Then choose Easy Medical Device. We can represent you and also become your importer. Contact us at eo@easymedicaldevice.com. So in terms of this, so we have some manufacturers that do some prototypes first, and then generation one, and then they do some tests and generation two. So and I suppose in between there are some modification, maybe of design, of material, of things. So this is mainly the things that can be also the problem here. So your advice is mainly to wait until the end, or to try even with the prototypes or with the generation one, because maybe it will guide you to the preferred, material to choose here.
In this case, we can see, and we have seen several strategies. You can develop a prototype and test this prototype as it will be the final form to guide you on did I choose the right material for my final device? Does my cleaning process leave residuals or not? And does my proto process, are they okay for biocompatibility? You can do this, but those tests won't be representative of the final device. However, if you perform correct and complete chemical characterization with collecting data, generated data by a chemical study for extractable and leachable, for example, on this prototype, when you will change of prototype, you pass to phase 2 or second edition of your device, which is not the final. You can carry out another chemical characterization to compare with your previous data. Okay. And maybe state on, okay, the new version, the V2, is safer. We use safer material, better non-biocompatible material, and the chemical characterization of leachable and extractable is safer, or I would say cleaner. And in this case, and it's often what we do here, what we can say and what we could try to do is to try to define a biological equivalence between V1 and V2. And if we succeed, meaning that the two versions are presenting the same risk, or the version 2 is less risky than the previous one, we can use data on a riskier medical device to evaluate the cleaner, the new one. And by using tests in the step of your biological evaluation, you perform tests on each step of the development of your device. You will be able to maybe use those R&D data to confirm the biocompatibility of your final device without testing the final device. But it requires a lot of work, and to justify everything you do, to notify, to keep all data you will generate during the development of your device. It is possible, but it has a cost also.
Exactly.
Perform tests each time you have a new version, and maybe it's not the right strategy for each company.
Exactly. No, I think it's also a good strategy maybe to mention that using data from different prototypes and the history of this can be also helpful also for you to build first the right product, so that we will have no issue, and then also to prove that you are making some progress on your products, and finding also, as you mentioned, the worst case maybe, so for the testing also.
Yes.
So you
are documenting your development as well. The design of your device is documented by using those data.
Exactly. Okay. Next issues.
Next issues will be to choose not choosing the right condition when you perform a testing.
Okay.
You have done your evaluation, you have done your chemical characterization, and now you're aware that some tests are needed. We will use the final device. It's easier. The final device is the final one. It is sterilized. It is clean. It is the device the patient will encounter. And now you have to think about the condition in which this device will be tested in biocompatibility testing. People tend to here at ICARE to give us the choice of the condition, because we had more experience sometimes than the client we have, and we are focused on the solvent you will use if you do an extraction, the animal, if it's an animal study, which animal you will use, and if you can avoid testing animals. That's a really important point. We at ICARE perform in vitro testing, alternative testing, and also in vivo, but we tend, and all the industry tend, to reduce this type of testing. So it's important to, when a client comes and say to us, I want to do an irritation test, now we have the rights and we have the standard confirmation that for medical device, you can do in vitro testing. So first step will be, as we can see this device, can it be tested in vitro test? That's the first question. Then, if you have to evaluate a more complex device, and that's imply implantation, some really big studies that can last for years and cost also to the sponsor, we tend to adapt the condition to the clinical use of the device. It's really important that your condition of testing are as close as possible as the clinical use of your device. This means if you have contact with specific part of the body, for example, the nose, the mouth, genital or reproductive organs, we can adapt a test to implant or to make in contact with a device and the subject at this point precisely to be most relevant as possible for the evaluation.
So I suppose that in that case, you have also, for example, when you receive products from customers that to be evaluated, you have also a set of questions to say which condition it is used, how it is used, etc., so that you can also adapt your test for this kind of things.
It's maybe the first question we ask to our client. What is the contact? Where your device will be used, and what will be the contact patient device? And maybe also the regulatory document we use in our GLP laboratory. The GLP procedure obliges requires us to do this. We have to check all this point: type of contact, duration of contact, specific type of contact. It's really important. If you have another example, but okay.
If you have a medical device in indirect contact with your patient, for example, for the extraction process, or earlier I was saying that you take your medical device, you place it in a vial, and you fill it with an extraction solvent. But for this type of medical device, sometimes filling is not the appropriate way to be representative of the clinical use, because the contact is only passing through I don't have an example of an indirect...
a tube or a catheter that will not directly contact the patient, but the body fluid, blood or something else can pass through the tube and then contact the patient. You will not perform tests in direct contact with cells or any animals. You will perform an extract by passing your solvent through the tube, like it will be used in clinical condition. Yes, that's one of the most common example we have. Sometimes clients are coming with tests. Yes, we test the catheter in direct contact with the cells to test its cytotoxicity. The tube was cytotoxic. Yes, because the external part of the tube is not intended to contact any viable cells or tissue, and it's covered with a plasticizer, which made it shiny and and nice. But the part that interests us is inside. So we have to test the inside of the tube and not the exterior.
No, it makes completely sense, and I suppose it's obvious now. But when people are thinking, they're thinking, okay, let's use it like that. But you have really to understand how it is used, so that you can really understand, as you said, to maybe use some solvent to remove some of those leachables or extractables from the interior of the tube, so that you can really test that part and not test the external that is never touching. Because the contact indirect contact or direct contact is also something that is important. I know that we have worked on that, for example, on the tubes for dialysis or this kind of thing, where the blood is going inside and then going back to the body. So yeah, the fact is that it's the interior of the tube that is in contact with the blood and then goes back to the body. So it's that part that should be tested. Yeah.
And it made me think of a thing. Sometimes clients test for when they have a test using extraction for a tube, like we did just now. They put the tube in the solvent and extract some stuff that won't be in contact with the patient. And for us, it's often difficult to interpret those results because the real part of interest is completely fluid in the other chemicals released by the external part, and this led to maybe too much testing because we cannot correctly assess the risk, and so we tell to our clients, you have to do all the tests because it's completely a mess in the analysis. We cannot do anything. And when we tell us you can just let the solvent pass through the tube, they said to us, yes, but the standard do not stand that we can do this. Yes, but the standard gives you example, gives you a way to follow. If you go a little bit on the side of the way, as long as you justify, and I think as you prove that what you've done is in favor of the safety of your patient, of your device, it will always be accepted.
But it's also common sense that should be used here. So it's I know that the standards are maybe not perfect, but at a certain point you have also to use some of your common sense for that. So okay, next issue.
The fact that biological assessment is not a one-day process.
Yeah, I mean it's a good one, this one, because everyone say, oh, can you accelerate that? Can you make it shorter? Can you? I will pay more, but give it shorter. I mean, it's not
yeah, and the fact that once it's performed, you can use it until you can use it forever, but you cannot. You have to know that your biological assessment is something you can do and we can do at one time. But it will evolve. It has to evolve with regulation, the MDR, with updates of standards, and even these updates on this regulation are based on what are evolving in our society. For example, we were talking before the MDR of nanoparticles, of CMR substances, of endocrine disruptors. And then now they are in the MDR, and you have to assess that in your biological assessment. So it will not be a frozen time process. Apart from these regulation issues, it will also evolve if you change your raw material in your medical device, if your supplier change, because a small modification in your medical device can lead to a big important change for the patients in the end. So
I suppose also, as you said, if your supplier change, mainly you have to redo maybe some of those tests, or you have to justify why you should not redo some of those tests, etc. So there are a lot of those things. So I suppose also that maybe it's better when there is a change of supplier. This should be already on the plan to make this testing before to really implement the change or to really buy any of a new product. So, and here the duration of the test should also be taken into account to say, it may be a 6 weeks process before you can get an approval for that. So you should not think it's just a one-day process and then you will be moving forward. Okay, is there any other issues here? I think we are
maybe two little points. This is not mistakes, but it's a point that often manufacturers are not aware of, and we prefer to talk about it here, so the people will know. Some endpoints in the famous table of the 10993-1 are not check in the table.
Yeah.
It's a case for degradation and reproductive toxicity and developmental toxicity. Does not mean you don't have to evaluate this point. You have to consider it if your device may be induce those risks or may degrade into the body. That's point. The more and more we see feedback from notified body asking to manufacturer of medical device, did you assess the potential for toxicity to reproduction or the potential of degradation of the device? And the client said to us, I'm not using my device in any sensitive population for this type of toxicity. It is not intended to degrade into the body. Yes, of course, but you have to tell and to show that you consider this endpoint and that there is nothing to do if your device do not degrade, if the targeted population are not pregnant women or really, really young child. You have many other ways than testing to prove that you have considered this endpoint and you have managed the risk associated to this endpoint.
I think it's always this. It seems obvious for the manufacturer, but you always have to have a justification why this is fine or why this is. As we said, the auditor will ask you, have you done this or have you done that? You can say to him, oh, it's obvious because of this or that. And you say, okay, where is it written? Where have you justified that? Do you have a document that proves that you have considered this case and that you have decided not to do because of that? And this is mainly the thing that, as you said, the specific test that should be done. We think that no, it's not needed, but at the end, you have again to have a justification or a document that is explaining that.
And then we add to this point that we have many clients that come to us with when we ask what is the targeted population for your device, and they say everyone. Oh, okay. All means. Newborn babies, means old people, mean immunodepressed people, mean pregnant women, mean everything. And when we tell, no, just adults, okay, be precise. If you tell us just adults, we will perform evaluation focusing on this population, general adult population with any problem, without any problem of health, without any disease. If you are producing a medical device specifically for pregnant women, that can occur, okay, we will focus on toxicity to development for the infant, for the newly born babies. We will focus on this point. But often the clients, yes, it's all population. All population means child and adults. But be aware of tiny population, tiny sensitive population. We often forget and need to consider in the evaluation.
So it's important for you to have all those details so that you can adapt the test to be done.
And once again, we can, with the most data, the most precise data we have, the less tests will be performed for the client.
Just maybe to emphasize on that. So, have you had already some customers that were not maybe specialized on all those things, and they are just coming? Here is my device, test it for whatever you want, and give me the results, so that it's fine. So, do you have some kind of those kind of manufacturers?
It happens. It's rare, but it happens. And in this case, it's my favorite client because we have to explain us, we have to educate us, like you said earlier, on the importance of biological evaluation and explain to the client that we are not here to take your money and tell you to go do testing. No, we are here to follow the regulation, make the document we provide to the client in compliance with the regulation, in order that the client will never come back to us asking for refund or asking for correction. We are here to guide if the person don't know nothing on the regulation. It's a pleasure for us to explain and to initiate them to the medical device world.
No, it's great. The last mistake?
What was it? Yeah, it's again, not really a mistake, but a really important point. It's where you want to put your device on the market. We talk about MDR in this podcast, which is the European regulation for medical device. But Europe is not the only market in the world.
Exactly.
And many of our clients want to go to USA, to North America, where the FDA is the authority that puts your device on the market. Both markets are, how we say, guided by guidance on the biological evaluation. Standard ISO 10993 apply in Europe and in the USA as well. But the FDA often goes beyond the recommendation of the standards. I would say the FDA is often tougher than the Europe to accept medical device biological evaluation. And thankfully, FDA provide to all manufacturers and to everyone a guidance, a document, which called the FDA guidance on ISO 10993.
Okay.
And this guidance, updated in 2020, gives the specificity of FDA. For example, they ask for specific condition of extraction when you perform a biocompatibility testing or chemical characterization. Using 3 solvents instead of 2 that are accepted in Europe. Some tests must be performed on using a specific methodologies. In cytotoxicity, for example, they require XTT tests rather than blue tripon tests that are often used in Europe. Some technical differences that we are aware and some specialists are aware. But the FDA gives a guidance on that, and I think when you can give the link to this document to the people listening to the podcast. It's a free document published by FDA. There is no.
Sure, let's put that in the show notes and that people can get that. But yeah, you're right that there are some specific requirements. So if somebody comes to you without telling you that maybe this is a biological evaluation for this country or that country, maybe those tests results will not be accepted by those countries, I suppose.
So that's also an important point to tell us when you first meet with us, when we start working together, is where are you going to put your device on the market? Is the USA? Is it Europe? Is it Japan? Is it all the continent have their own regulation? We can follow, of course, we can follow the standard of ISO that available all around the world, but some countries have specificities and you have to be aware of this.
Yeah,
and
sure.
We have to know it to guide you as well as we can.
No, it's true. I know that, for example, for other elements, for other testing, they have different standards, they have different temperatures. For example, sterilization. In the US, the sterilization process is different than in Europe, etc. All the guidance are different. So you have to do the testing for the different temperatures, the different timing, the different things. So yeah, it's true that this is also something that is important. So don't hesitate to go, if you go to Groupe ICARE or to any other laboratories, to have an understanding of what is really required. I mean, I hope that within all those mistakes that we talked about, people have now an understanding of what exactly they should provide as an information, so that they can save some time, be efficient, maybe save some money also, because mainly, this is the idea also to make this process really efficient. And for that, so Groupe ICARE, what else are you doing? I mean, apart from doing biological evaluation, what other services you can offer for the customers?
So before beginning any biological safety assessment process, we can guide manufacturers throughout this process. We can also write biological evaluation reports that will compile every data we talked about in this podcast in a report that will conclude on the safety of the medical device. We can analyze chemical characterization data, which means that we can analyze data you already have, collection of data, and we can analyze data you generate through chemical characterization or biocompatibility testing. We can perform worst cases on if you have a range of several medical devices and you want to establish which medical device can be the example of all your range to be assessed, we can do that as well.
We can do anything.
Yeah, it's already a good list here. So mainly this is something that a lot of manufacturers are needing, and yeah, as we talked last time. So yeah, Groupe ICARE, you are not only for biological evaluation, you are also working in the different field like sterilization, like testing, like. Do you have also, as we talk about biological evaluation, are you doing also the test on animals also directly?
Groupe ICARE perform most of the tests required by ISO 10993-1 in the table. Most of the tests are done. We have an in vitro lab who performs cytotoxicity, irritation on episkin, so reconstituted human skin, all type of gynotoxicity, hemocompatibility. And on the other hand, we have in vivo laboratories when we can perform irritation, sensitization, systemic toxicity of any duration, acute to chronic, and implantation. We have a train team who can perform implantation in rabbits or in rats, depending on which type of device you want to evaluate.
No, it's great. I mean, I hope people will be contacting you directly when there is any need for that. And we talked before to register this podcast. We talked about also a webinar that you are planning. So maybe you can tell us more about that.
So we talk about medical device here, but ICARE provide evaluation and provide services in other domains such as chemistry. We are planning for the 21st of February a webinar on the chemicals and alternative testing. So how you can do your regulatory test on your chemicals without using animals as required by the European regulation, and as it's also also the case in medical device. So the next webinar will be on chemicals and alternative testing, and I think no, I don't want to say a mistake on who will present the
yeah, no problem. I mean, I will put you send me also a link, and I will put that on the show notes, and then people can go directly and register if they really want to.
It's free. It's about 30 minutes of presentation and 30 minutes of question with the viewers. So go ahead, check it if you're interested.
Great. Okay, so thank you very much. I hope all the information that we provided was helpful. And as I've said, you can see now that Groupe ICARE has also the right people to help you to perform this kind of process and help you for all the biological evaluation. So don't hesitate to contact them. I will put all the information and the details on the show notes for the profiles of Laura and Paul, but also for the website of Groupe ICARE website. Yeah. Okay, Laura, Paul. So really thank you for your help and the information, and I wish you a nice day.
Thank you, Monir. Bye.
Bye-bye.